So today we're going to be talking about the digestive system and digestive system disorders. But before we get into that, we're going to talk about the macromolecules of life, which I just mentioned. We never really got a chance to talk about macromolecules of life in A&P1. We went straight to cells and cell biology in A&P1. We kind of ran out of time. So all macromolecules of life are proteins, fats, carbohydrates, and nucleic acids. And I've been using that terminology all along. I just want to solidify that idea before we go into the GI tract, because we will be talking about disorders of digestion and absorption. And guess what we're absorbing and metabolizing is proteins, fats, carbohydrates, and nucleic acids. That's why this lecture is here today and not anywhere else. So again, usually if you're doing a full anatomy course or you're doing a human biology course, you would start all the way down from atoms and you would talk about how bonds were formed to create molecules. When we say macromolecules of life, we mean that there are 4 molecules, macromolecules, large molecules, that all of your cells are made out of these 4 types of molecules. Everything that is you from head to toe is made of these 4 kinds of molecules, plus vitamins and minerals. That's literally all you are, is these 4 things. So it's nice to know what you're made out of, right? When we say digestion and absorption, we say we break down large molecules into small molecules. We're breaking down polymers into monomers. We're going from this. We're going from a complex chain of things, a complex build of things. We break them down into a single piece of that thing. And then in our cells, we take those into our cells and we build that into a component of the cell. That's what we're saying. That's what I've been saying all along. I'm just putting it into one single sentence for you and one single phrase for you. So that single building block for all of our macromolecules, that's going to be, the general term for that is monomer. So monomer, mono as in one. One unit of anything is a monomer. And for our different macromolecules of life, they have different monomers. And when we build up, I'll finish this thought and then I'll answer your question. When we start to build them up, we create bonds between monomers, like putting Legos together. We build something up, we call that a polymer. Poly is in many. Any more than 2 units, that's a polymer. Okay, what's your question?
You said a polymer turns into a monomer and then the monomer goes to build into the components of the cell?
Exactly. So we're going to go from polymer that you ate, break it down into a monomer so it can cross the digestive epithelium. Then it's going to be transported to your cells, and then they'll metabolize them. They'll build them into new polymers. So like you had a Lego castle, you broke it down, you turned it into a Lego X-Wing instead. Does that work for everyone? Okay. So that's our general terminology again, polymers versus monomers. So part of your responsibility for this segment is to know what the monomer is for each macromolecule of life and what some of the polymer forms are, some of the terminology associated with monomers and polymers for each of the 4 macromolecules of life. That's a big part of what we're doing here. So let's go back to just the monomer for carbohydrates. And our general monomer for carbohydrates is glucose. To keep it really, really simple, just for simplifying, it's glucose. Glucose is made out of carbons and hydrogens with some extra oxygens in there compared to some of the other molecules we're going to see. It usually takes the form of a ring. So every time that I've illustrated this as a hexagon, that was not incidental. It's hexagonal in nature. There's a couple of different forms of glucose. Honestly, alpha glucose is the one we can eat. Beta glucose we can't. Very, very small changes in these molecules, like the direction of this is called a hydroxyl group. This hydroxyl group going down means that we can break apart the polymer into glucose. This hydroxyl group being oriented in this direction means we can't. It means there's an enzyme that can work with this. There's no enzyme that we have that can work with that. That's how specific enzymes can be. Beta glucose is found in cellulose, so like plant cell walls. When you eat vegetables, you're getting dietary fiber. That's the same thing. That's cellulose. It means you can't physically break it down. You're not getting energy from it. You are getting dietary fiber, and that's going to help you poop. Yep, talking about poop. Beta glucose is going to be the monomer for fiber. We're really focusing on alpha glucose. That's the one that's going to be that energy source for creating ATP. Okay. So again, glucose is the monomer. Carbohydrates, there are very different kinds of carbohydrates, such as cellulose and starch. Those would all be considered polymers. Starch being the one you can digest, cellulose being the one you can't. And of course, we can turn this into all kinds of things that are not specifically about energy sources, but we don't talk about them as much, so I'm going to move on from that. Fatty acids are the building blocks of lipids. It's hard to say that there's a monomer exactly for this. What a fatty acid is a chain of carbon backbones. We're a carbon-based species. Most everything is carbon. If you don't have a letter there, it's a carbon in chemistry. These are all carbons. So we're still talking about a carbon chain, just like this was actually a carbon chain that turned into a loop. And then we're going to have hydrogens, just hydrogens pretty much, bound to that carbon chain. So this would be a fatty acid. We would still call it a fatty acid if it was a shorter chain. We would still call it a fatty acid if it was a larger chain. So saying that it's a monomer is a little bit weird for this molecule, but we're going to roll with it. There's a ton of terminology that gets co-opted by sort of fad diets. And you will get this in chemistry and you will get this in nutrition, but I just want to inform you of a couple of things that get co-opted by fad diets and really don't mean what the general public thinks they mean. So when we say a saturated fat, all we mean is saturated by hydrogen. That means for every carbon, there's 2 hydrogens. Carbon, 2 hydrogens, carbon, 2 hydrogens. That was what we mean by saturated fat. And the thing about this is that it lies flat. And form and function will be related. If it lies flat, then at room temperature, it's going to be solid. Just because it stacks neatly. That's the only reason. Now, an unsaturated fat means that there is a double bond somewhere in this chain. And that double bond creates a kink in the chain, just a physical kink in the chain. And that means it does not stack and it will not be solid at room temperature. Yes?
How come coconut oil is considered so good for you, but it's also saturated?
Excellent question. Coconut oil is a saturated fat, or at least it's 51% made of saturated fats, so we'll consider it a saturated fat. It's also medium chain fatty acids, so these chains are shorter. And what we say is the bioavailability of that as an energy source is higher than it is if the chain is longer, which is not at all what people are being taught in the general public about it. Yes.
So since the chain is shorter, it's easier for us to break down.
That's the thinking, yes. I'm simplifying, but that is accurate, yes. For medium chain fatty acids, MCFAs, is what you would see in the literature. Yep, exactly. Most of our saturated fats are like animal fats, like the fat that you trim off chicken and things like that. But coconut oil is a saturated fat as well. So an unsaturated fat, there's a double bond and that creates a kink. And because it no longer lies flat, it's no longer going to be solid at room temperature. It's now going to be liquid at room temperature. So your olive oil, your canola oil, things like that will be unsaturated fatty acids. And you can have multiple double bonds, and the more double bonds there are, the more kinks there are, the more liquid it's going to be at room temperature. It also makes it less stable, so there's a shorter shelf life in our unsaturated fats. And then you have cis and trans fats, which is another buzzword. All that means is cis means same, trans means across. Cis means same side. So this hydrogen is on the same side of the double bond. That double bond means it can't rotate. That means this is a cis fat. If that hydrogen was down here, it would be a trans fat. That's it. That's the whole thing. So saying that something is unhealthy because it's a trans fat, unhealthy because it's a saturated fat, healthy because it's an unsaturated fat isn't quite accurate. There's some correlative stuff where trans fats tend to be highly processed, and there are some bad things associated with that, but all it means is where the hydrogen is, literally, in a literal sense. So saying that something is not a trans fat doesn't make it healthy, is a better way to phrase that. Okay, so fatty acids, a number of things will be considered the polymer forms of this. You see lipids in your phospholipid bilayer of your plasma membranes. There's a bunch more. Let me go to this really quick, see if I've got a good example for fats. Triglycerides are a really common and significant polymer form of fatty acids. And that's it for fats. And a lot of that was just me giving you some very basic nutritional information. And the only significant thing to learn from this is polymer versus monomer. Any of these can be polymerized into different forms. So the monomer would be the fatty acid. The polymer would be like a triglyceride or a phospholipid bilayer element or any number of things. Okay, for your proteins, we're still starting with a carbon backbone. All of these started with a carbon backbone. That glucose had extra oxygen. Now a protein is going to have an extra nitrogen. We're going to go nitrogen, carbon, carbon, and then when we polymerize, it's going to go nitrogen, carbon, carbon again. That nitrogen has some clinical significance too. You know from your renal lecture that you're going to measure nitrogenous wastes when somebody's got a kidney disorder, right? We say if you've got one kidney, you have to control your protein intake because of nitrogenous waste buildup. You get nitrogenous waste when you eat meat. That's why you're limiting meat. When it's because you're limiting protein. You're limiting the amount of nitrogens that you're getting. So this is the amino group. This is the acid group. That's why this is an amino acid. And once again, we're just working with carbons and hydrogens, and now we've got extra nitrogens and oxygens involved. Then there's this thing right here. This is the R group, and we call this a functional group. It's actually just standing in because this is highly variable. You have a number of different amino acids that involve a number of different arrangements of molecules. They have different electronegativities. They interact with each other differently. That's going to be what makes proteins fold in certain patterns. They're going to have, I don't want to say magnetic forces because it's actually the opposite of that, but things that are attracted to each other or repulsed from each other based on what R group is in that specific amino acid. And you've heard of these phrases all along, right? You build proteins based on your genetic code. You take this specific amino acid, that specific amino acid. This is what we mean by a specific amino acid. Which R group does it have? Which functional group does it have? So again, that R group is going to determine how it's going to bond to other parts of the protein and then how it folds, and then it's going to fold into a shape, and that shape has a function. Yep.
One way I thought that Rachel described it well was thinking of this like a necklace, like it being straight or being curled up or doing all that, and that with this part.
Yeah, absolutely, like a necklace, and it's going to bunch in a certain way. Or maybe you could do like your headphone cable, and you put it in your pocket, and when it comes out, it's like a massive knot, and it just does that on its own. Yeah, you could do it that way too. Like that. Exactly. Perfect. Any questions about proteins? Hopefully this is all like clicking from different things that we've been saying all along. And you're just like, oh, proteins folds. Oh, nitrogenous wastes. Oh, carbon chains. Oh, hydrogenated. Like all of that stuff should just be going click, click, click. At least that's the goal. Last one is nucleic acids. That is our building block of DNA. So again, the monomer here is nucleic acid. The monomer is DNA, RNA. It's also ATP. It's our basis for ATP. So we actually have another sugar. Instead of it being a hexagonal glucose, it's a pentose sugar. It's still got a carbon backbone. We're still talking about mainly carbons and hydrogens and some oxygens here and there. We do have a nitrogenous base, so there will be nitrogen in our base pair. That should sound familiar too when I say base pair. I am talking about A to T, C to G. And then a phosphate group. So that phosphate has that electronegativity, the oxygens all around a P. Can't remember what that is. It's a phosphate group. You'll have these bound together in your genetic code, in your DNA double strand, double helix. It'll also be the basis for your adenosine triphosphate. So there's your adenosine and here's your triphosphate, 3 phosphate groups attached to it. So nucleic acids are also the monomer for ATP. Nucleic acids are also the basis for ATP. They're also the monomer for ATP, in addition to genes. So anything that you consume that was alive, any plant, any animal, that has nucleic acids. Generally, we don't need to worry about getting nucleic acids in our diet because we eat living things like plants and animals. If you're eating plants and you're eating plants, wheat is a plant, right? Greens are plants. Everything was plants at some point. Then you're getting nucleic acids. So we don't really worry about it as much in our diets compared to proteins. Again, proteins being the polymer, amino acids being the monomer, fatty acids and glucose are the ones that we really monitor. So there's our sort of representation of a glucose. This is, again, why we're seeing this here today is because we're going to have specific carrier molecules or carrier proteins or very specific transporters for these slightly larger molecules to get them across plasma membranes. It's not water, it can't do osmosis, it's not small and nonpolar, it can't simply diffuse through the plasma membrane. We're going to need specific carrier molecules. And again, those carrier molecules are going to have to work, right? So here's a nice summary slide for you showing you a polymer form of starch, a monomer of glucose, the DNA double helix and the monomer nucleic acid, a chain of amino acids that are part of a larger protein, and there's the monomer amino acid, and there's a fatty acid, and there's a triglyceride. Triglyceride being the storage form of fatty acids that you would find in your adipose cells, for example. Any questions about literally that was a 17-minute lecture on the macromolecules of life. That could honestly have been an entire semester to cover just that content. So questions, comments? Did anybody feel like some puzzle pieces came into place? Good. Yeah, what's up?
So if we go back to a couple of slides, these purple things are
Carrier proteins.
Are the proteins?
Yeah, they're going to be integral proteins of the plasma membrane. Carrier molecules are a transporter protein. They're going to change shape in response to the binding of a selective molecule. So this one would be a glucose transporter in particular.
So it would be like what's the difference between that and a glycoprotein?
A glycoprotein, so generally when we say glycoprotein, we mean an integral protein that's anchored in the plasma membrane, but has a strand of some kind of starchy strand, and its role is going to be really different. So the glycoproteins I know about, their role is to attract water and be hydrophilic. So things like glucosamine and chondroitin would be glycoproteins in your joint spaces that keep fluid in your joint space so that it's lubricated. Yeah, this is about transporting glucose across the plasma membrane. Yeah, so this is a carrier molecule. We'll see what these molecules actually are today. Well, I think it might be review content, but we'll look at it again anyway. They're actually going to work based on the sodium gradient. It's going to be a co-transporter with sodium for both amino acids and glucose. It's a sodium co-transporter. Okay, and here it is, an exact thing that I just told you about. When we're getting carbohydrates, we consume carbohydrates. Let's say that there was sugar in my tea. There's not, but let's pretend there was. I would consume that carbohydrate. My salivary amylase would begin the breakdown of any sugary molecules into monomers. And salivary amylase isn't going to do it on its own, but it's eventually going to get there. We're going to have mechanical breakdown, we're going to have chemical breakdown, and then we get to the small intestine where most of our absorption is going to occur, right? So far, so good? We have mechanical breakdown, that's digestion, that's a separate process from absorption. Are we all comfortable with that? Okay, absorption is when we're going to get our glucose molecules to cross the intestinal mucosa. We've got to get through this columnar cell here in the intestinal mucosa and into a capillary bed that's lining the gastrointestinal tract. So where are the capillary beds embedded? Where is your blood supply of your small intestine located? What's the structure that suspends your small mesenteries? Well done. So there's this capillary bed, the source of that would be from the mesenteries, and it's just going to be on the other side of that gastrointestinal epithelium. Okay, so what we have on this side of the intestinal mucosa, we have microvilli to increase surface area, and on those microvilli are going to be some co-transporters. So the co-transport is between sodium and glucose. So it's going to be coupled with sodium. We're going to assume you've got a lot of sodium coming in, because typically we do eat a lot of salt, and that there's going to be a lot more sodium here than there will be here. So the sodium gradient, that gradient triangle for sodium would look like this, right? We would have sodium really wanting to go this way. So we'll use that energy of the gradients to power through the glucose and push it into this cell. On this side, however, we'll just have facilitated diffusion. We're no longer going to use a sodium gradient. We will just use some a basic carrier molecule just for glucose to get it from this side of your simple columnar cell into the capillary bed. And the process for proteins is going to be pretty much identical. We're going to have facilitated diffusion with co-transport with sodium again on this side of the intestinal mucosa. This side, we're also going to have facilitated diffusion and co-transport, but it's still going into a capillary bed. The most different one is lipids. Lipids we break down into fatty acids. They're going to use simple diffusion when they're small and nonpolar enough. And then on this side of the intestinal epithelium, on the basilar surface, we're going to use exocytosis. And they're going to get into lacteals. What's a lacteal? It's your lymphatic system, right? Not your lymph nodes. So we're going to transport lipids via the lymphatic system, not by the cardiovascular system, is all we're saying here. And that chyle now, right? Now we're going to call it chyle, that fluid that's in the lacteal, that'll be transported to the liver. All of these capillary beds are going to be transporting things to the liver, including the lacteals are going to go to the liver. And that's going to give you your first pass metabolism. Have you talked about that in Pharm? Tell me a little bit about first pass metabolism. Yeah. Right. Right.
So is it like more like you're breaking down the enteric coating?
So you'd break down the enteric coating in your stomach, for example. But yeah, you're going to be absorbing it across your small intestine epithelium. And then again, if it's is it a fat-soluble drug? Is it a water-soluble drug? Is it going to be transported via lacteals? Is it going to be transported via the cardiovascular system? Either way, it is going to the liver, and the liver gets the first shot at it before it's circulated to the rest of the body. So it is what you said. I'm just clarifying it, and also for the recording so they can hear it. Okay, that was our review. Do you guys feel comfortable about digestive enzymes, breaking things down, building things up, how things are going when they're working properly? Okay, let's get into some gastrointestinal disorders. We're the organization of this overall is that we're going to go from mouth to anus in order, more or less, if you want to think of it that way. Delightful, I know. Just makes sense. We do use the terminology proximal and distal in the GI tract just to orient because the actual small intestine is wiggling all over the place, right? So proximal would be towards the mouth, distal would be towards the anus, just for that simplicity's sake. Usually we'd only use that for appendicular, but it works for GI. So we're going to go from proximal to distal if we want a more pleasant way to say that. So talking about butts. So we'll start with this concept of dysphagia. Dysphagia is not a disorder in and of itself. It's terminology that we will refer to for a number of things that make it difficult to swallow. If somebody's having difficulty swallowing for some reason, we'll simply refer to that as dysphagia. It could be an inability to initiate swallowing. There could be some kind of sensation that makes swallowing unpleasant and something that that person is going to try to avoid, or it could be that it's actually painful. So here are some major causes of dysphagia. These aren't the only causes of dysphagia, but this is a good selection. And again, it's an image, but you still need to know everything on here. What's up?
Is dysphagia only the pain and difficulty, or is this also when they don't like to swallow and it goes in the wrong place?
It would be difficulty swallowing. If it's going to the wrong place, I imagine they might be having difficulty swallowing, but I'm not entirely sure. You would call it dysphagia. It's still dysphagia.
Right. Okay. Oh.
Gotcha. So it is applied to, even if it's not difficult, it's going in the wrong place, and they're going to avoid it anyway, though, right? Yeah. If something is causing a person to not want to swallow, would be another way to phrase that. So one cause of dysphagia would be fibrosis. If there's some kind of scarring in the esophagus, then that could narrow the esophagus and make swallowing more physically difficult and unpleasant. Again, this is going to be visceral innervation, which means what? What's up with visceral versus parietal innervation? What's the difference between your body wall's innervation and your visceral innervation? Yeah, so it's innervation of an organ versus innervation of the body wall. So let's say you had an ulcer in your small intestine. Can you point to that part of your small intestine and say, here's where my ulcer is? No, but if you get stabbed in your body, can you point to where you got stabbed? Probably. So visceral innervation is really nonspecific. They may follow referred pain patterns. It's going to be vague, a gnawing pain. It's going to be described differently than somatic or parietal pain, where you have dermatomes that give you a very specific body map of where things are, right? That's visceral versus parietal, or somatic rather. So the pain of dysphagia will tend to be very nonspecific because this is visceral innervation. Another one would be a tumor. If there's some kind of compression, including but not limited to tumors, let's say there was an enlarged lymph node in this region, maybe there was a lymphoma, it's a tumor of a lymph node growing to compress the esophagus. A diverticulum, you can have diverticula that are congenital, like you were talking about swallowing and it's going to the wrong place. There can be a congenital form of that, or it can be a result of scar tissue buildup. And remember, we're talking about walls in the GI tract, or layers and tunics, just like we did all throughout AMP2. There's tunics, there's layers. A layer can separate away as a result of trauma and scar tissue, and then that can develop into a diverticulum. And now food's going to go into the diverticulum. Sometimes when somebody says, I just feel like things are sticking in my throat, things are literally sticking in their throat. Yeah, and then you've got a nice little rotting bolus right there. Yeah, that's a problem. Let me zoom in on this. I think it's atresia, but I want to make sure I'm getting the word right. Congenital atresia. Remember your embryology, where you're a series of tubes on tubes on tubes at a certain stage. You do have a gut tube from mouth to anus. It's all one tube, and it is possible to have a lack of connection between one part of the tube and another. There's also development of fistulas. You learned from embryology last week that the lungs form as a bud off of the gut tube. That is part of that endoderm development. That was part of our embryology last week. So there is a congenital way to have what's called a tracheoesophageal fistula. It just means it came from the esophagus and it kept that connection to the esophagus rather than developing the full septum. This is neurological. This is autonomic visceral innervation. So if there was a neurological disorder impacting some of your nerves of digestion, such as, I mean, what's your major parasympathetic rest and digest cranial nerve? Vagus. It's always vagus, right? It's always vagus. So if you had some kind of, let's say, medulla oblongata damage, that's very likely to damage vagus and very likely to damage your gastrointestinal peristalsis. And then achalasia is also going to be about peristalsis. So it's going to be, instead of having the rhythmic contraction all the way down the esophagus, at some point, either it's chronically tightened. In all likelihood, this would be like peristalsis is chronically tightened, and you're not going to be able to get past that. It's going to make a sort of sphincter, like a tetanic sphincter in a way, before it can empty into the stomach. So once again, that would be a different way of describing I feel like food's getting stuck in my throat or in my esophagus. Sometimes it is. What's that? Stroke patients? Absolutely. Yeah, loss of peristalsis. Loss of peristalsis can happen anywhere along the GI tract. For dysphagia, it would be esophageal, but we will see, for example, megacolon later, and that's going to be a lack of peristalsis in the large intestine. That makes sense. That makes a lot of sense. For the sake of the recording, she just said that patients who've had a stroke have to pass a swallow test before they're given any kind of food. Makes sense. Okay, again, because this is visceral innervation, esophageal pain very often is going to express as heartburn. It's going to be referred to as heartburn, pyrosis. One cause of esophageal pain or heartburn would be reflux of gastric contents, esophageal reflux. We will talk about GERD a little bit later, gastroesophageal reflux disorder, in which that epithelium is actually going to be changed. But right now we're just talking about manifestations of dysphagia and esophageal pain. Another thing, what's up? Oh my gosh, yeah, let's look at the physics of that real quick. I think this is actually going to come up later in the lecture. Let me make sure I'm sharing my screen with you really quick. No. Oh, that's great. Exactly, yeah. There's a really great video if I can find it, and it's absolutely terrifying. Oh, I can't find it. Oh, that's not it. There's a lot of terrible ones, apparently. Oh well, I'll look for it later on break. You're right. It's because the baby's growing and it's going to grow in the way that pushes organs in the superior direction. You're really just not going to have room for all of the organs in your peritoneum anymore. Yeah, super fun. Sounds delightful. Yeah, sounds about right. And that's 20 out of 40. So by the end of it, we'll actually talk about how you can have not just reflux, but a herniation of part of the stomach in some cases through the esophageal hiatus and into the mediastinum in some cases, as a response to that baby literally physically pushing in that direction. Yeah. I say yes. I have a lot of body horror around pregnancy and childbirth. It sounds terrible to me, but if you talk to Karen about it, she'll be like, baby. So, okay, heartburn. Da, 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 da. Another source of esophageal pain and dysphagia, for that matter, diffuse esophageal spasm. This runs in my family, so I get to look forward to it. This is discoordinated peristalsis. It could also be caused by infections or problems from immunocompromised patients, but as I understand it, it can be genetic as well because it runs in my family. Hooray. So what you're looking at is a barium swallow of the esophagus, and all of the peristalsis is happening at random rather than happening in a coordinated fashion. This is why you learn peristalsis in AMP2. Again, that's going to cause some manifestations such as angina pectoris, pain to the neck, shoulder, arm, and jaw, and swallowing worsens that sensation. So as the women in my family get older, they tend to avoid eating certain things. And my mom tells a story of her aunt who would look at the kids eating and she would just be like really angry and look mad at the kids for being able to eat and she can't eat. Yeah, I would be mad. Just being bitter. We already talked about this, but remember that there's a difference between visceral pain and somatic visceral being poorly localized, gnawing, burning, cramping. And somatic pain is going to be more sharp, tense, well localized. You may also experience referred pain patterns from visceral pain, and that means that our pattern of innervation for visceral nerves, it's not going to be as specific and well aligned as the dermatomes are. There may be ascension or descension before that joins up with a spinal nerve, and then along those pain pathways there may be crosstalk. So while this is the part that's experiencing inflammation in the organs, that neuron firing is going to activate the same neuron as a patch of skin in a dermatome. And that's how we're going to get some of our wacky referred pain patterns. So this one, for example, inflammation or pain or experience of death of cells on the heart may radiate to the pectoral region or perhaps the left arm if you are male. If you're female, however, you could feel nauseous, you could have back pain, jaw pain, neck pain, fatigue, tired. Basically the way you feel right now. Yeah, and that's one of many reasons that women have worse outcomes for heart attacks than men. Men know, I'm touching my arm, I'm having a heart attack, get me to the ER. Women are going to go, I feel like this most of the time. I'm just going to ignore it. Take some Tums, I'll be good, it's fine. Yeah, there really needs to be another PSA for the fact that women have a different referred pain pattern than men for heart attacks. Let's see, I think we're good there. Any questions? Some basic overview stuff, constipation. Does anybody have enough radiology under their belt where that visually makes sense and is immediately terrifying? It is. It's impacted fecalith. So that's the head of a femur. That's the acetabulum of the coxal bone. Head of the femur, acetabulum of the coxal bone, fecal impaction. All of that. Yeah, like basically it's the size of a baby's head. It's the size of an infant at that point. Yeah, like a full-term baby right there of stool. What's that?
I just said that's like a small child.
Small child, yeah. No. Okay. Lots of causes for this. So constipation is small, infrequent, or difficult bowel movements. Again, there could be a number of different reasons for this. It could be related to an obstruction. It could be neurological in source. It could be metabolic in source. It could also be from a lack of exercise. Just increasing your motility will affect your intestinal motility. Dietary, low in fiber. Get that indigestible cellulose into your system. Highly recommended. Here in Colorado, there is a major issue for just hydration. My cadaver has had dementia with failure to thrive was her cause of death, and she had a fecal impaction of about that size. And it was just because we're in Colorado and she was constantly dehydrated and she had dementia and couldn't regulate her water intake very effectively. So as I, worked on her, I learned that that's very common in Colorado. All I know about it is that opioids decrease intestinal motility. I can't tell you what the mechanism of action for that is. Ask Ashley, she'll know. I mean, I know a little bit about it, but you know, they're opioid receptor agonists. It was a long time ago, so that's a good Ashley Watson question. And then, of course, the opposite of constipation is going to be diarrhea, increased frequency and fluidity of bowel movements from any number of causes. And here's some causes. Anybody Invader Zim? No? Yeah, good stuff. So diarrhea could be because of osmosis. If you have some indigestible solute in your large intestine, where does water go? To stuff. So osmotic diarrhea would mean that there's some poorly absorbed solute in your intestines and water is going towards it, and that's going to cause diarrhea. So when I say poorly absorbed solutes, that could be any number of, what's that? Yeah, you do it on purpose with some of your laxatives, right? But it could also be because of a short gut system syndrome, or maybe we've surgically removed part of the small intestine and you've decreased absorptive surface. So all of that stuff that should have been absorbed throughout the GI tract has not had enough time. That would be another cause of that. What's up? I don't know. It's a great question. Let's try to find out over break if there's some plastic thing that can happen. Secretory and exudative, there's going to be a bit of a gray area here where if you look at different sources, they'll put different things. Both of these are going to be related to, so this one's going to be infectious inflammatory. But this one's going to be due to toxins, but a lot of those toxins are going to be from infectious things. That's where I think the gray area comes from. I almost don't really see a reason to separate these two categories necessarily, but your textbook is separating these categories. So anything that would be toxin, it doesn't necessarily have to be infectious to be toxic to you. We could put it that way if you really want to differentiate these things. And then motility disturbances, like I mentioned, that would be neurological for the most part. So exudative diarrhea, again, that's this category right here. C. diff would be a major source for this. A number of different bacteria, but also some inflammatory bowel diseases like Crohn's and ulcerative colitis. We will talk about inflammatory bowel diseases in just a bit. It's past time for a break, and that's a good stopping point.